introduction. My contribution to you all today is to share my experiences primarily as a cell biologist, but also as someone who has dedicated his own career, as Anne said, to the field of biomaterials, in particularly biomimetic biomaterials. And today, indeed, I would like to share what has been our journey when biomimetics, or as other people call it, biomimicry, can be basically pushed down to the nanoscale level, and to do so to control the phenotype of the cells, and moreover to direct them towards the formation of tissue-like structures in vitro. What here in the title is called organotypic cell culture. So today, what I would like is, without patronizing anybody, I know that there are cell biologists among you, we would like to explore very quickly what are the biochemical and cellular pathways controlling the formation of these tissue-like structures, and then look at a novel class of biomaterials called phenodrive that is commercialized by TissueClick, and as that indeed that property I was referring to earlier on, to mimic the microenvironment of the cells at the nanoscale level. And I hope to be able to give you some examples about how this material is operator friendly, enables reproducible results, the experiments, and basically can be applied both to the culture of stem cells, particularly adult mesenchymal stem cells, but also induced pluripotent stem cells and tissue cells in general. And I will give you just a range of potential of cases that where we have applied these biomaterials to different types of cells to see, to show pretty much the potential of its use. So in terms of introduction, as I said, without patronizing anybody, but just for the sake of tuning our brains on the same wavelength, I would like just to remind you that the cells, obviously, the cell phenotype relies very much so on the ability of the cells to recognize its own extracellular environment, and that this interaction is basically obtained through receptors, and these receptors do not only guarantee a physical grip for the cells with the surrounding environment, but their interaction with specific bioligands presented to the receptors by the macromolecules of the extracellular matrix can indeed affect intracellular pathways and eventually gene expression, dictating the behavior of the cell. One of the important features that we want to consider today is that the presence of these ligands, like the 3 amino acid sequence, very well known RGD sequence, is also important for another aspect of the interaction, which is the clustering of integrins across the phospholipid bilayer, which again, as I said before, can control intracellular pathways. Another important aspect for us to consider today is that among the different types of substrate extracellular matrices that we find in the body, the stem cells have a particular environment which is called the stem cell niche. And in this niche, cells can form 3D spheroid-like structures, can undergo both symmetric and asymmetric proliferation, but all the stem cell niches, although different from each other depending on which type of tissue they are located in, they have some common denominators. And these common denominators for our own interest today is the basement membrane. The basement membrane, as we know, is made of different macromolecules, but today I would like to bring to your attention two in particular. The collagen type 4, which unlike other types of collagens that, as we know, have a fibrillar structure, collagen type 4 has a mesh-like structure. And then there are other molecules, and among which laminins are very important for the presentation to the cells of those bioligands necessary to the recognition through integrins. If we switch now from what is happening in nature to the way we culture cells, we are all familiar with some attempts that we all make as cell biologists to avoid the interaction of the cells with the tissue culture plastic. And therefore, we have adopted various substrates like matrigels, or in general collagen-based substrates, laminin, fibronectin, polylysin, polyornithine, to control indeed the cell phenotype more than in the case of the culturing on tissue culture plastic. If you look at those proteins that I've listed before, most of the proteins that I've listed in the previous slides, and as I said before, they all have some specific amino acid sequences that are recognized by cell receptors. I'm not going to go through this very busy slide, busy table. It's just here to say that there are a number of peptides that can be synthesized pretty easily in the lab and used to provide substrates, synthetic substrate for cell culture, rather than relying on the batch-to-batch variability of natural and expensive protein substrates. Now, the problem of using these receptors in terms of what I said before, in terms of controlling the integrin interaction and the integrin clustering, is that there is no chemistry available to us in first place, at least not any of the conventional one, to make sure that these receptors here indicated with these red rods or blue rods can be presented in a way that is controlled both in terms of density at the surface and spacing, controlled spacing, which we believe is important for the clustering of integrin. And there are examples of gels that have been proposed and put on the market for cell culture, like Pura Matrix, that originally didn't have any of those functionalizations, self-assembling hydrogel that didn't have those peptides exposed, but that more recent works have shown to be able to be functionalized with those peptides to improve cell adhesion. Yet, also in this case, obtaining a controlled density and controlled spacing, moreover, is very difficult. So all of this comes at a time where, as I said, biologists, we all need the more and more to orient our work towards organotypic cell culture. The so-called organoid platforms, organ-on-chip or body-on-chip, relies indeed on the ability of controlling the cell phenotype A and B, to be able, obviously, to form tissue-like structure on our tissue culture plate or on a chip, as shown in this diagrammatic picture on the right-hand side. And that's where PhenoDrive, the product currently produced by TissueClick and delivered by PELOBiotech, comes in. PhenoDrive is not just one product, but rather is a range of products that can be sold as a freeze-dried powder. This freeze-dried powder can be easily dissolved in any aqueous or alcohol ethanol medium, in particular at a given concentration. In particular, PhenoDrive, in our case, as you will see later in the data in the slides where I show our data, is dissolved in a mixture of ethanol water, is pipetted into the well at a given volume, and then let to evaporate to cast a very transparent, invisible film in our tissue culture plate, obviously under sterile conditions. After a gentle washing with other PBS or the same tissue culture medium used during the experiment, cells can be seeded on PhenoDrive. And the technology is based in a nutshell on this, on what you can see in these diagrammatic pictures, whereby PhenoDrive is based on the functionalization of various linear polymers that are functionalized with a hyperbranched structure. Now imagine like a nanotree where you have the root of the tree being covalently bound, grafted to the linear polymer, and the uppermost branching generation are those red rods that you have seen in the previous slides, which can present to the cell membrane and indeed to the cell receptors those bioligands in a very controlled manner, both in terms of density, depending on the number of branches, and in terms of spacing, again, depending on the number of branches. So basically, in this case, for example, you have that the bioligands presented to the cells are sequences like one of the laminin or the common RGD that I have mentioned before. Now, if you look at the PhenoDrive from a physicochemical characterization point of view, and you try to we remind ourselves what I said before about the basement membrane, where collagen type 4 has a mesh-like structure, and laminins are able to present those bioligands we have discussed so far. You can see in a nutshell in these slides the two being confirmed. In terms of density, if you look at a linear polylysin that has been functionalized with any of those peptides, and you compare it with PhenoDrive, you can appreciate the higher density of the bioligands presented to the cells for the same square area compared to the polylysin. If you do scanning electron microscopy and you look at the surface that the cells see when they sit on PhenoDrive, you can see that the mesh-like structure similar to those of collagen type 4. So in a nutshell, with PhenoDrive, we can mimic at the same time the mesh-like structure of collagen type 4 while presenting to the cells in a very controlled, high-density manner bioligands like those of laminins, but as I said, not only. We had the first go at proving the potential of these substrates by looking at the effect on the mesenchymal stem cells. Here, as you can see, stem cells form 3D-like spheroids on a 2D tissue culture plate coated by PhenoDrive. And you can appreciate that basically these results were obtained when each of the nanotrees that I've shown in a schematic way in a few slides before were presenting 16 branches exposing the laminin sequence YIGSR to show how important the clustering of the integrin and the spacing of the YIGSR or any other peptide is in terms of cell response. We have repeated this experiment not with branched molecules exposing 16 branches, but rather 8. When you do that, you can see that the cells, rather than forming spheroids, go back to the more traditional spread-like structure and colony-forming units to which we are used to. And then obviously, without going too much into the details because of the time available, we did studies, first of all, to prove that these cells were maintaining their pluripotency, looking at nanog and OCT4 transcriptor factors, but not only those. We looked also at the most traditional one, like CD75, CD90, and etc. for mesenchymal stem cells. We wanted to look at the way integrins were basically localized in these spheroids, and you can see here that, as you would expect, integrins were expressed only by the cells that were at the periphery of the spheroids, rather than by the cells inside the spheroids. And interestingly enough, and you will see a nice video in the next slides, that the cells that were sitting at the periphery of the spheroids were expressing N-cadherin, which is a marker of cell migration. And then we wanted to understand why they were forming spheroids. And in a nutshell, we found out that the spheroids were expressing higher, overexpressing the Rho factor, which is known to inhibit the formation of the cytoskeleton of the cells, therefore inhibiting the spreading and rather encouraging the formation of spheroids. And this video shows how the spheroids form over 3 days. So we see that the cells in a serum-free tissue culture medium. You can add, after a few minutes, the serum if you wish, changing the medium. But we saw that the cells can survive up to 7 days in serum-free conditions. And you can see how cells are migrating towards each other to form spheroids. And you may notice that during this migration, the philopodia of the cells are looking for some kind of vesicles left behind by other cells. This was not happening, obviously, on tissue culture plastic and matrix gel, as we demonstrated on PhenoDrive. We looked at more carefully, we're still studying, and we hope to publish soon results to explain this migration mechanism. And we here you can see that the stem cells during migration is blabbing, is forming and releasing matrix vesicles, which are deposited on PhenoDrive and reached out by the cell philopodia. And to prove that these were matrix vesicles, we have done staining with integrin, considering obviously that those vesicles could have be fragments of the cell membrane, but we are now conducting more specific study to characterize these vesicles. And here you can see another beautiful example of the formed spheroid. Another interesting aspect was to look at the paracrine effect of these stem cells when assembled into spheroids. What we did was to culture, to do the experiment you have just seen, and then take the supernatant of those cells and use it to spike endothelial cells sitting on a matrix gel and look at the angiogenesis potential, the tubuli-like formation. As you can see, in the case of supernatants coming from the spheroids compared from supernatants coming from stem cells growing on tissue culture plastic and polylysin, you can see basically how a more structured angiogenic tubuli are formed by UVEC, by umbilical endothelial cells. And we established that this was due to a higher production of vascular endothelial growth factor by the spheroids. So we moved a step forward and we say, can we make our culture condition more complex, trying to mimic the perivascular stem cell niche? And here you can see another paper we published 2 years ago, whereby we compared Matrigel with Phenodrive for UVEC only. So as I said before, the tissue culture plastic was coated either with Matrigel or Phenodrive. Cells were seeded in serum-free conditions, and you can see how more structured the angiogenic tubuli are formed by UVEC when they grow on Phenodrive like on Matrigel. More importantly, as we know, Matrigel was degrading after 4 days in culture, where we could keep Phenodrive for at least 15 days, 15 days. And then we co-cultured the UVEC with the mesenchymal bone marrow, mesenchymal human stem cells. And you can see that in the case of Matrigel, the stem cells, here stained in green, tried to form interaction with the tubuli of the formed by the endothelial cells. Whereas in the case of Matrigel, you have a much more ordered structure where spheroids of stem cells were forming, particularly mainly at the anastomotic junction of the network of tubuli. We wanted to understand the mechanism of sprouting of endothelial cells on Matrigel and Phenodrive. And without going too much into the details, you can see how, in the case of Phenodrive, the tip cells that form the angiogenic sprouting as its integrin clustered in a puntiform manner across its perimeter, whereas in the case of Matrigel is more homogeneously spread. We looked also at why, in case of Phenodrive, cells were forming spheroids more at the anastomotic junction, and we attributed this process to the clustering of the high density expression by the endothelial cells of connexin 43 in the anastomotic junction. Connexin 43, as I'm sure you know, is a receptor that cell receptor that endothelial cells use to establish connection with other types of cells. I'm not going into the details of this slide, where we looked again at the formation, reactivation of ROA that you have familiarized with before. Rather, I want to give you reassurance that these spheroids on the stem cells were keeping higher level of pluripotency as determined by the expression of this factor of pluripotency of adult mesenchymal stem cells. And again, we did other studies looking at other transcription factors, but in the interest of time, I leave it to your reading. This is an open access paper that you can find online. Also, the paracrine ability of these receptors of these stem cells and endothelial cells was looked at for different receptors, for different growth factors in different circumstances. Again, this is too much to go through into details today. Rather, I want to show you another example of how Phenodrive can be used. This is not stem cell. These are not stem cells anymore, but they are cell line hepatocytes, the typical HPG2 that are co-cultured on Phenodrive together with UVEC. As you can see here in the beautiful scanning electron microscopy picture at low magnification on your right-hand side, high magnification in the center, you can see that the hepatocytes were forming lobuli-like structure like the one we find in histology in the liver. But moreover, these spheroid lobuli-like structure were connected by either relatively large tubuli-like structure, which eventually could branch into very small capillary branches all around the lobuli-like structure. We use this, and we have not published, we hope to publish this data soon, to look at the biodistribution of nanoparticles which is an important issue in the study of new nanomedicines. So we treated this spheroid, we spiked these spheroids with different types of nanoparticles in the context of an European project. And we found out that the nanoparticles had the tendency to interact more with the endothelial sprouting rather than the endothelial, hepatocytes themselves. So if you think that nowadays, most of the all of studies of biodistribution, which means accumulation of nanoparticles for toxicity study, are conducted in animals, these types of models give you the opportunity to study it at the cellular level, obviously without ruling out animal models, but giving you a different level of information. We did the same and we published paper where endothelial cells were mixed with beta cell, human cell line, beta cell line. This is a type of human pancreatic beta cells that is very argued in literature because they don't produce insulin. Very interestingly, we were able to use PhenoDrive in two conditions here. One is what we are now familiar with, the coating of tissue culture plastic, where we had basically the endothelial cells forming the sprouting and the pancreatic cells forming a spheroids-like structure on the top of these tubuli-like structures. But here we also published data where the two types of cells were mixed with PhenoDrive in suspension under rotary conditions. Obviously, in this case, PhenoDrive is not dissolved in ethanol, but it is dissolved in the tissue culture medium at the very low concentration, 0.1 weight volume, and under normal rotary conditions, or if you would like, we also have tested it in a microgravity reactor. The cells can form, as you can see on the right-hand side, very beautiful structures, whereby the green staining cells are the ones positive to markers of beta cells, whereas the endothelial cells here again were forming sprouting, angiogenic sprouting in a 3D space. More interestingly, we did a characterization of the connexin 43 again, as you have seen before, in different normoglycemic, hyperglycemic conditions. But what is the most of interest to you is that both the typical marker of expression of beta cells were highly expressed in PhenoDrive, and I would say that in this case, PhenoDrive was combined to gelatin as main linear polymer to be functionalized. But the most important aspect of our work was when we compared the production of insulin in normoglycemic and hyperglycemic conditions. As I said before, this type of cell line pancreatic beta cells do not produce normally insulin in tissue culture plastic. Here we demonstrated that when assembled with endothelial cells in the form of spheroids, they could produce insulin and moreover react to hyperglycemic stimuli. Again, this is an open access paper that you can find available. I want to finish with the last example of my presentation today, looking in a nutshell at a study that was not conducted by my team, but by our colleague at Phenocell France. Phenocell is a company selling induced pluripotent stem cells. What the colleagues at Phenocell did in the context of European project that we had with them was to compare PhenoDrive with polyornithine laminin to understand whether the two substrates could induce the differentiation of induced pluripotent stem cells in neuron and astrocyte of the cortex layer number 4 of the brain, which is important for study of neurodegenerative conditions. Here you can see beautiful staining almost equivalent of a phenodrive with polyornithine laminin, which is the common substrate used to differentiate induced pluripotent stem cells into this type of cells. These markers of expression that you can see here shown in a microscopy slide were also quantified by our colleague at PhenoCells, showing all positive results in terms of maturation of differentiation, of induced pluripotent stem cells, either into into neurons or astrocytes, depending on the conditions. I'm not going to go too much into the details of studies we have conducted in vivo, where phenodrive was used to coat medical implants like dental implants, showing the formation of high-density bone trabecula in the jaw of the sheep. But here we had a functional group exposed by the branched molecule, which was phosphoserine, which is known to be one of the most potent catalysts of biomineralization in the living kingdom, from mollusks to humans. And here you can see how quickly the surface was mineralizing in presence of phenodrive. And also we included phenodrive in injectable material for 3D printing, which was eventually injected also in a rabbit model, showing a higher accelerated regeneration of bone and of differentiation of stem cells into osteoblasts. Last but not least, we looked at the opportunity to use phenodrive to functionalize the porosity of collagen scaffolds. Here there is a commercially available product used for osteochondral defect regeneration by Finceramica Italy, whereby one layer of collagen is mineralized, the other layer is non-mineralized. We looked at the functionalization of these materials with phenodrive, and you can appreciate the difference. Here there are stem cells adhering on the control non-functionalized materials, where cells go into spreading as you would expect, whereas in the case of phenodrive you have spheroids that are gradually merging into larger structure, and in vivo model in sheep you can appreciate the beautiful regeneration not only of the cartilage but also of the underlying bone. So I want to conclude my talk saying that biomimetic materials nanoscale, or if you like, synthetic extracellular matrix analog, can be produced in a very reproducible way. They are operator friendly, and moreover can drive our studies to the level of organotypic cell culture, where cells can indeed form tissue-like structures in a different condition, applied to testing, as I briefly shown, on nanoparticles, but also drugs and other types of testing of biomaterials. We have also the opportunity, and the time today was not enough to show you some work we have done where phenodrive has been added to bioinks for 3D printing, again giving the similar results that you have seen before, in the 3D collagen scaffolds applied to osteochondral defect. I want to finish acknowledging all the funding bodies that have supported our research in the UK, the UKRI under the form of the EPSRC and BBSRC, but also the European Commission that for many years has supported our projects, and most of the data you have seen today have been achieved through those projects. And we are also very keen and proud of having a lot of quite few early career research young scientists who have progressed from University of Brighton. They moved to TissueClick, and some of them have now moved to very brilliant careers in a larger company or academic institution. And with that, I would like to thank you for your kind attention.
Thank you very much, Matteo. So if you have any... Was? Du hörst mich nicht?
Can you hear me, Anne?
Yes, I can.
Okay.
Yes, I can hear you.
I mute my mic in case there is some feedback noise.
You still can't hear me? Yes, now? Okay, perfect. Yes, thank you so much, Matteo. So we already have a few questions. If anyone else would like to ask a question, please use the chat function. The first question would be, what are the cell types working best so far with PhenoDrive?
Thank you. As I shown in my presentation was kind of a chronological discovery journey that we did on PhenoDrive. So definitely there is an advantage for using PhenoDrive for mesenchymal stem cells, maintaining their pluripotency. But we didn't have time to present also very interesting data on the lung cells and satellite cells that, as you know, are the stem cells of the skeletal muscles. So basically, I would say that stem cells, but any type of cell line, has shown this tendency to form organotypic structures, particularly when co-cultured with endothelial cells. And so on the priority list, I would put not only stem cells, but also endothelial cells, because as I've demonstrated, I hope today, the sprouting that you can have with the UVEC is much more consolidated than matrigel in first place, but also is also more stable, so you can run your experiment for much longer period of time without encountering any problem. With the UVEC, the cells were surviving easily in serum-free medium and forming the sprouting that you have seen for up to 7 days.
Okay, wonderful. Is the sound working again, Matteo? Do you hear me again?
Yeah, please go ahead. Yes.
Okay, okay, wonderful. So that would probably lead to what is the most common application of PhenoDrive users?
I would suggest it as a coating of the tissue culture plastic, simply because the advantages are quite few. First of all, the fact that you form such a thin layer, which you don't see by naked eye, which has a very good impact in terms of quality of the microscopy. So you, unlike other gels that obviously can produce problems also of autofluorescence when you do immunostaining, with PhenoDrive you have none of that. So that's one advantage when you use PhenoDrive as a coating of tissue culture plastic. The other is the stability over time. Easy to coat the plastic, is very easy, operator friendly. So these are the three main advantages. If you do studies where it's required to grow cells in absence of serum, again, we have demonstrated that up to 7 days, most of the cells survive. So I think that there are advantages not only in terms of the behavior of the cells, but also to facilitate the operator in the preparation, culturing, and eventually characterization of the cells with either light microscopy or confocal microscopy, immunofluorescent microscopy in general.
Thank you very much. So the next question that we have would be whether you have done or do you know of work done with primary human hepatocytes versus HepG2?
The answer is no, and it's a very good question because many people have asked me the questions in previous seminar webinars. One of the reasons why we didn't use it is because the work was done in the context of European project where we had to test nanomedicine, and it was the choice of the consortium to make those tests that we were meant to develop during the project to be accessible worldwide for and in a reproducible way. So we decided to do all our studies on cell lines, but indeed, I think that it would be very interesting to repeat the experiment on primary hepatocytes. And to be honest, I don't expect difference because we have not done hepatocytes, but we have done other types of primary cells and showing very good performances in terms of maintaining the phenotype of the cells and forming organotypic structures.
Perfect. Thank you very much for that question. And another one just came in. Have the biophysical and structural properties of the substrate, stiffness and nanotopography been analyzed? And if so, can they be controlled?
So the nanotopography, I showed my very first slide where I showed the scanning electron microscopy showing the controlled nanotopography, the nanotexture produced by PhenoDrive when it is used as a coating for tissue culture plastic. So that has been done. In terms of stiffness, again, that's a very good question. I think we have not done it, but it depends. In my view, it may depend, in my view, from the type of linear polymer that we use and the thickness of the coating. So the answer is we haven't done it, but I would suspect that one of the advantages that can be controlled. In fact, another advantage of PhenoDrive is the fact that you can use it as I said, invisible thin layer, but you can use also it in form of a gel. So TissueClick can provide different formulation of PhenoDrives, not only in terms of bioligands presented to the cells, but also in terms of kind of physicochemical properties. And for the 3D printing, when PhenoDrive has been used as additive of bioink, we have demonstrated that there is no change of the injectability rheological properties. So referring to the mechanical properties, the stiffness, the softness, I can expect that the PhenoDrive can produce those effects on the cells from the point of view of presentation of bioligands without changing what are the original mechanical properties of the materials used and functionalized with PhenoDrive. Can you hear me, Anne?
Yes, I can hear you.
No, okay.
Oh yeah, there we go. So thank you so much, Matteo. So thanks for also answering all these questions, and of course, thanks for everybody for asking. And I guess our last question would be, what will you try next? Is there anything that you have in the pipeline?
Yeah, I mentioned briefly during my presentation, we are very curious to understand the mechanism of migration of stem cells of PhenoDrive looking at those vesicles. So we have at the moment some members of our research team at the Center for Regenerative Medicine and Devices looking at a characterization of those vesicles. And then we are having very, very interesting data when we compare PhenoDrive with the laminin, collagen, fibronectin, looking at the way that satellite mast cells can differentiate to form myotube-like formations. With our data so far show that with the polylysine, which is recommended by the supplier of the stem cell or the satellite cells, the satellite cells just maintain their phenotype and they keep proliferating. Similar things happens to collagen fibronectin. However, when we seed the cells, we culture the cells on PhenoDrive and laminins, they tend to form this fiber-like structure, like the myotubes of the striated muscle. So we are now focusing on that aspect of the work, which is very challenging, but very, very nice challenge to take. And also we are looking at breast cancer cells because we are developing some gene delivery system for breast cancer, and we use PhenoDrive as a model to study the mimicking of the tumoral mass and the metastasis formation because of the migration that the PhenoDrive can encourage.
All right, so it sounds like you won't get bored anytime soon.
Not really, no, we're very, very happy bunny. After the punishment of COVID where we couldn't get in our labs, now we are full mode in.
I know, yes, you're right. There was one thing that just came in, Matteo.
Yes.
The question, would you be able to supply custom PhenoDrive products with a custom peptide attached?
That's a very, very good question because the answer is yes, and it's one of the things that the PhenoDrive class of product offers. So the answer is definitely yes. The only problem could be if that sequence that the customer wants us to provide them with is too long. In that case, you may have some problem with the steric hindrance during the synthesis because the peptide is not grafted on the hyperbranch structure, but is synthesized alongside the structure. So if the sequence is not too long, let's say no more than 6-7 amino acids, definitely it is possible to be done.
Perfect. Thank you very much, Matteo.
Thank you all.
So, yeah, I guess that would be all the questions that people asked. And for me, there's only left to say thank you for your talk today. Thank you for answering all these questions. This session has been recorded. If anyone would like to go back and look at it again, you can do so on our website. Let us know if you have any more comments or questions. And then thank you, Matteo. Have a wonderful day. Hopefully not too hot. And I hope to talk to you soon.
Thank you for having me, and have a good day, everybody.
Bye. Thank you, everybody, for registering.
Bye.
Bye.